When an animal intakes food with an unusual substance, it may trigger an allergic reaction. Once that substance of the foreign antigen combines with the blood circulation, the IgA will detect the foreign antigen, and this will result to the binding with the mast cell. IgA mast cell complex receptor will bind to the foreign antigen that will initiate the release of vasoactive molecules, enzymes, cytokines, and chemotactic molecules that would cause acute inflammation and systemic effect, which is associated with the release of histamine that causes systemic vasodilation. A dog was submitted to a breeder instead of a doctor. The dog had wounds and injury. The breeder injects penicillin. After a while, the dog shows hypersensitivity reactions. Inside the blood vessel, there are albumins that are component of blood. They are not antigenic since they are small molecules. When the penicillin is inside the blood vessels, it will bind to the albumin, making it antigenic. Antibody and complements will detect its antigenicity and will opsonize it and let the natural killer cells apoptosize. One of the typical manifestations of type 3 hypersensitivity includes Arthur's reaction. In this case, the dog was given repeated subcutaneous injection of antigen that leads into formation of immune complexes.
Dexter. Ooh, wait. Pick it up, pick it up, pick it up, rude. Baby girl, watch how you move. What? I got them vents on my shoes. I'll pop up, put it out.
Antibody complex, also known as immune complex, attached to the endothelium of the blood vessel wall. The complex attached to the tissue activates the complement cascades beginning with C1, which leads to the activation of protein C5a and C3b. C5a acts as a chemotactic factor for neutrophils and attracts them to the site. Upon binding, the neutrophils release cytotoxic granules containing reactive oxygen species that damage the body tissues. In case of Arthur's reactions, this leads into visible edema, severe hemorrhage, and followed by ulceration. When tuberculin is injected into an animal infected with mycobacteria, a delayed hypersensitivity response occurs. In these animals, a red indurated swelling develops at the injection site. The inflammation begins after 12 to 24 hours, reaches its greatest intensity by 24 and 72 hours, and may persist for several weeks before fading gradually. When tuberculin is injected intradermally, it is taken up by Langerhans cells, which then migrate to the draining lymph node. They present antigen to memory T cells that respond by generating Th1 effector cells. The circulating Th1 cells recognize the antigen when they encounter it in the skin and accumulate around the antigen deposit. The T cells recruit other Th1 and macrophage to the site. The macrophage also releases serotonin and chemokines. These chemokines attracts basophil. Basophil-derived serotonin or histamine cause yet more inflammation and enhance migration of mononuclear cells into the lesions.